So our last topic to cover in the nervous system is the autonomic nervous system. Now, when we look back at this slide that we've seen before, the basic structure of the overall nervous system, we find that the autonomic nervous system is a branch of the peripheral nervous system that controls visceral function. So the targets of the autonomic nervous system include smooth muscle that lines the vasculature and also lines the viscera. It controls the function of cardiac muscle, and it also controls the function of both exocrine and endocrine glands. Okay. Now, the autonomic nervous system has two arms. It has the sympathetic nervous system, which we think of as being our fight or flight response, and it is active or activates our stress-related responses. And then the other branch is called the parasympathetic nervous system, which is thought of as our rest and digest response, which is actually the sort of baseline function for the autonomic nervous system. So the parasympathetic nervous system maintains resting conditions, maintains baseline function, and then under stressful conditions, the sympathetic nervous system turns on. So this is describing rest and digest versus fight or flight is really describing the efferent arm of the autonomic nervous system. But just keep in mind that there is also an afferent arm to the autonomic nervous system. There are this is sensory information that comes in, this is visceral stimuli that comes into the spinal cord and then up to the brain, and it is relaying to the brain the baseline conditions, or rather the ongoing conditions of visceral function. It conveys information such as blood pressure, heart rate, respiratory rate, and so forth, okay, and a variety of other parameters. There is a basic organization that we see to the autonomic nervous system, and this is true for both the sympathetic nervous system and the parasympathetic nervous system. Okay? And this basic organization is really talking about the efferent arm of our autonomic nervous system. And so the basic organization, if we simplify it very much, we describe it as being a system composed of two cells and two synapses. So the first cell has a cell body that resides within the central nervous system. We call this cell the preganglionic neuron. Okay? It conveys information into the peripheral nervous system and synapses onto a second cell that we call the postganglionic neuron. Okay? And the postganglionic neuron has a cell body that sits outside of the central nervous system, okay, somewhere in what we call a peripheral ganglion. And a ganglion is really just a collection of cell bodies, synapses. Okay? There are even sometimes some axon fibers that run through a ganglion, but it's really just a collection of cell bodies and synapses that are sitting outside of the central nervous system. And that's what a peripheral ganglion is. So the cell body of the postganglionic neuron sits in one of these ganglia, and it projects and synapses onto ultimately our target cell. Okay? And these are the target cells include what we just mentioned: smooth muscle cells, cardiac cells, exocrine, endocrine, glandular cells. Okay? So two cells, two synapses. Okay? And this is the basic organizational structure of both the sympathetic and parasympathetic nervous system. So we're going to talk about the details of each system, starting with the sympathetic nervous system. The sympathetic nervous system, so we're looking at a more detailed description of our basic two cells, two synapse arrangement. Okay? So we said the preganglionic neuron, or the first cell, its cell body is sitting in the central nervous system. So for the sympathetic nervous system, those cell bodies are actually sitting within the spinal cord, okay? And they are sitting in the spinal cord starting at level thoracic level 1, and it runs until thoracic level 1 to 3. Okay? There's a little variation whether it ends at thoracic level 1 or ends at thoracic level 3. Okay? And when we say that the cell bodies of the preganglionic cells, okay, for the sympathetic system, is sitting in the spinal cord, if we jump here for a moment, we see that the cell bodies, all the cell bodies in the spinal cord sit in the gray matter, right? We mentioned that. And we kind of ignored the lateral horn when we talked about the gray matter before, because the lateral horn is where we find autonomic cell bodies. Okay? And so for the sympathetic nervous system, the cell bodies of the preganglionic neurons are going to be sitting in the lateral horn of the spinal cord between levels T1 and L3. Okay. And we also saw this picture before when we talked about the structure and function of the peripheral nervous system. We talked about the ventral roots, the ventral surface of the spinal cord and the ventral roots that come out, and the dorsal surface and the dorsal roots that come out of the spinal cord, and that they merge to form spinal nerves, right, that are a combination of both sensory and motor fibers. So if you notice here, down here, what we have is branching out of the spinal nerves is this structure here, okay, these fibers that lead to this structure that runs lengthwise down the vertebral column. Okay? And there are swellings at every segment of the spinal cord, of the vertebral column. This structure is called the sympathetic chain ganglion. Okay? So the preganglionic cell bodies of the sympathetic nervous system are sitting in the lateral horn of the spinal cord, and they're projecting out, okay? And they have to make that first synapse, onto postganglionic neurons. And so for many of these preganglionic cells, that first synapse is going to happen at this structure, the structure called the sympathetic chain ganglion. Okay? It is literally a chain of ganglia that run on both sides, along both sides of the spinal cord, of the vertebral column. Okay? And because it runs on both sides near the vertebral column, we call these ganglia the paravertebral ganglia. Okay? Para meaning around. Okay? So the sympathetic chain is where many of these preganglionic neurons are making their first synapse onto the postganglionic cell. Okay? There are some exceptions. For some preganglionic cells, they will pass through the chain without making that first synapse, and instead they will make the first synapse at one of these, what we call prevertebral ganglia. Okay? And in this slide, we're showing two examples, the celiac ganglion and the hypogastric plexus. So these are sympathetic ganglia that are distal or sitting away from the sympathetic chain, where some preganglionic cells are making that first synapse onto postganglionic cells. Okay? And then the postganglionic cells then extend and synapse onto target cells in visceral organs throughout our body, okay? In our autonomic targets throughout our body. So still talking about two cells, two synapse arrangement. First cell originating in the spinal cord, the lateral horn of the spinal cord between T1 and L3. Okay? That preganglionic cell, that first cell, leaves the spinal cord, and it's going to make the synapse either within the sympathetic chain, or it's going to pass through the chain and make that first synapse at the prevertebral ganglia, the celiac or the hypogastric plexus. Okay? And then that second cell, the postganglionic cell, is then going to extend out and make that last synapse onto the target cell. That's the basic structure of the sympathetic nervous system. There's one exception within the sympathetic nervous system to this two-cell, two-synapse arrangement, and that is what we describe as the endocrine portion or endocrine arm of the sympathetic nervous system. And that's where preganglionic neurons of the sympathetic system extend and directly innervate the adrenal medulla. Okay? This is the inner portion of the adrenal gland, which is an endocrine gland. There's 2 endocrine glands that sit on top of each kidney. Okay? And if we look at this here, this is the adrenal gland, okay, sitting on top of the kidney. So a preganglionic neuron is going to extend out of the spinal cord and directly innervate the adrenal medulla. And the adrenal medulla is going to release a hormone, epinephrine. Okay? The adrenal medulla is going to release epinephrine into the bloodstream. Okay? And epinephrine is going to then stimulate the target cells. Okay? So that's the one exception to this 2-cell, 2-synapse arrangement in the sympathetic nervous system. Okay? And that is the endocrine arm involving the adrenal medulla and the release of epinephrine. So the parasympathetic nervous system is different in its arrangement. Okay? It's very different in its arrangement. We still have 2 cells, 2 synapses. Okay? We still have that basic structure, but the location of these 2 cells and their cell bodies it looks very different compared to the sympathetic nervous system. In the parasympathetic nervous system, the preganglionic cells, the cell bodies of the first neuron, is sitting in, some of them are sitting within the brain. In particular, they're sitting in the midbrain and the medulla. So the cell bodies are in the midbrain and the medulla. Okay? And they are extending out of the brain through cranial nerves. There are specific cranial nerves that are composed of preganglionic fibers of the parasympathetic nervous system. Those cranial nerves include cranial nerve 3, 7, 9, and 10. And I need you to memorize, I need you to know those cranial nerves, that they are really parasympathetic. Okay, they are part of the parasympathetic nervous system. Their cell bodies are sitting in the midbrain and the medulla, and the preganglionic fibers are extending out of the brain through cranial nerve 3, 7, 9, and 10. And of course, cranial nerve 10 we talked about being the vagus nerve, and the vagus nerve conveys, it's an extremely important nerve for the parasympathetic nervous system. We have 2 on each side, okay? And the vagus nerve conveys, or the 2 vagus nerves convey, about 70% of the parasympathetic nervous system to our entire body. So you can see that the vagus nerve branches many, many times, and it makes multiple targets, innervations onto all of our viscera, or practically all of our viscera. Okay? The other location, the other place where we find preganglionic cells of the parasympathetic nervous system is within the spinal cord, in the spinal cord at level T12 to L1. So in the lateral horn of the spinal cord at T12 to L1, we find some preganglionic cell bodies of the parasympathetic nervous system. Those preganglionic neurons leave the vertebral column within the sacral portion of the vertebral column, between sacral level 2 through 4. Okay? And those preganglionic neurons are ultimately going to affect the function of some pelvic viscera, including bladder function and anal sphincter function. Okay? So we've got preganglionic neurons coming out of the brain, the midbrain and the medulla, coming out of, originating at T12 through L1 and leaving the vertebral column through sacral levels 2 through 4. Okay? And unlike the sympathetic nervous system, where there were very large and obvious peripheral ganglia where that first synapse occurs, in the parasympathetic nervous system, those first synapses are occurring at much smaller plexi that are scattered throughout the body. And they are located very, very close to and sometimes on the target organ itself. So that means that the postganglionic neurons are really very, very small, and they don't need to extend very far because they're going directly into the target organ. Okay. And an example of one of these peripheral plexi is the cardiac plexus, which actually is located near the aortic arch of the heart, where the aortic arch that extends out of the heart. Okay, so the cardiac plexus is a collection, is where the preganglionic neurons are synapsing onto postganglionic fibers that then immediately can innervate the heart. Okay. I'm going to talk about the neurotransmitters that we find within the sympathetic and parasympathetic nervous system, what receptors those neurotransmitters bind to, and then also a little bit more about the general arrangement of the entire autonomic nervous system. So there are two basic neurotransmitters that we find utilized by the autonomic nervous system. The first one is acetylcholine, and any neuron that releases acetylcholine or a receptor that binds to acetylcholine, we describe those structures as being cholinergic. Okay? The other neurotransmitter that predominates in the autonomic nervous system is norepinephrine. And any neuron that releases norepinephrine or receptor that binds to it, we refer to as adrenergic. So if we look at the sympathetic versus the parasympathetic nervous system, so here in this slide, what we have is on the very top is the somatic system, okay? And then underneath that we have all of the autonomic nervous system, starting with the parasympathetic system followed by the sympathetic system. And what's described here is the structures that we find in the central nervous system, in the peripheral system, and then the target organ. Okay, so at the target organ level. So if we focus our attention here on just the autonomic nervous system, we find that within the parasympathetic system and the sympathetic system, we have this two-cell, two-synapse arrangement. So everything's organized in terms of that. And it involves a preganglionic neuron that synapses in some sort of peripheral ganglion onto a postganglionic neuron, which then synapses onto the target organ. Okay? Now, for the parasympathetic nervous system, excuse me, the preganglionic neuron is synapsing in one of these peripheral plexi or ganglii, and the neurotransmitter being released is acetylcholine. Okay? So for the parasympathetic nervous system, preganglionic neurons release acetylcholine. And they bind to a particular subtype of the acetylcholine receptor found on the postganglionic cell. This subtype is called the nicotinic acetylcholine receptor. And it's called nicotinic because it was discovered that nicotine binds to and stimulates these receptors with equal strength to acetylcholine. So nicotine is what we describe as an agonist for this receptor. Okay? And that's not significant to us, except that it is the basis for the naming of the receptor. Okay? And then at the second synapse, the postganglionic neuron again releases acetylcholine, but it binds to a different subtype of the acetylcholine receptor. It binds to the muscarinic acetylcholine receptor, okay, on the target organ. Okay. And it's called the muscarinic acetylcholine receptor because a chemical called muscarine happens to be the agonist for this particular subtype of the receptor. If we look at the sympathetic system, we find that for the first synapse, okay, between the preganglionic neuron and the postganglionic cell, the first synapse is very similar. It's actually identical to the first synapse in the parasympathetic nervous system. So the preganglionic neurons of the sympathetic system release acetylcholine and bind to nicotinic acetylcholine receptors. Okay? And so that's true when the preganglionic neuron is synapsing onto the postganglionic neuron. It's also true when we look at the preganglionic neuron synapsing onto cells of the adrenal medulla. Okay? Releasing acetylcholine, binding to nicotinic acetylcholine receptor. Where we see the big difference is in the second synapse of the sympathetic nervous system. The postganglionic cells of the sympathetic system release norepinephrine. And norepinephrine binds to adrenergic receptors, okay? And there are different subtypes of the adrenergic receptor. There's what we call the alpha-1 receptor and the beta-1 and beta-2 receptors. Okay, so norepinephrine is released by the postganglionic cell, binds to an alpha-1 or beta-1 or beta-2 adrenergic receptor. The adrenal medulla is releasing epinephrine into the bloodstream. Right? This was the endocrine arm of the sympathetic nervous system. Now, epinephrine, when it gets into the bloodstream, it has a higher affinity to bind to beta receptors. So beta-1 and beta-2 receptors, epinephrine tends to bind to on the target cell. Okay. So if we look at these different adrenergic receptors, the receptors that bind norepinephrine and epinephrine, we find that alpha-1, beta-1, beta-2 tend to be located in different places. So alpha-1 receptors, which happen to not be on this list, alpha-1 receptors are the receptors that we predominantly see on the smooth muscle of the vasculature, especially the vasculature leading to or perfusing the viscera, our internal organs. Okay. Beta-1 receptors are adrenergic receptors that are associated with cardiac muscle. Okay. So they tend to cause an increase in heart rate, specifically cardioacceleration, or increase in cardiac contraction strength. Okay. Beta-2 receptors we find on structures like the heart. So I'm sorry, not the heart, but the lungs. So we see beta-2 receptors along the smooth muscle of the bronchioles in our lungs. And beta-2 receptors cause dilation of those structures. So we get dilation, bronchodilation, increased ventilation of the lungs. Okay. We also see beta-2 receptors in places like places like the bladder. Okay? And so when epinephrine or norepinephrine binds to beta-2 receptors, predominantly we're talking about epinephrine binding to the beta-2 receptors, the bladder will relax. Right? The wall of the bladder, smooth muscle along the wall of the bladder relaxes. Okay? The alpha receptors that are listed in this slide here is specifically the alpha-1 receptor, and as it shows, it's causing vasoconstriction when it's found on smooth muscle of vasculature. It mediates also the dilation of the iris, intestinal relaxation. Okay, it contracts the sphincter within the bladder. And one thing needs to be mentioned before we move away from this slide, and that's, while I said that the alpha-1 receptors are found on the smooth muscle of the vasculature or the blood vessels that perfuse our internal organs, okay, the smooth muscle lining blood vessels that are perfusing skeletal muscle, okay, tend to have beta-2 receptors, not alpha-1 receptors. And when epinephrine, norepinephrine binds to beta-2 receptors, it actually causes vasodilation. So within the context of a sympathetic response, we have norepinephrine and epinephrine binding to receptors throughout the blood vessels. Okay? And to the blood vessels that are sending blood to our internal organs, we see vasoconstriction or constriction of those vessels. When we're looking at the blood vessels that are providing blood to the skeletal muscle, we see a dilation, a vasodilation of those vessels. Okay? And so that's consistent with the idea of a fight or flight response. We're decreasing blood flow to our internal organs, increasing blood flow to our skeletal muscle so that we can either fight, either we can fight or we can flee. Okay? So that's an important thing to mention. So if we look at the targets of innervation for both the parasympathetic system, which is drawn on the right, the sympathetic system, which is drawn on the left, the targets are usually the same. Okay? So both systems usually target the same organs. Okay? And there's some exceptions. For example, the kidney is only innervated by the sympathetic nervous system, and there's no parasympathetic innervation to the kidney. But when both systems when both systems are innervating the same target, they usually have antagonistic effects on that target. So for example, in the heart, sympathetic stimulation to the heart increases heart rate, parasympathetic stimulation to the heart decreases heart rate. So they usually, both systems usually innervate the same targets. They tend to have antagonistic effects on those, or opposing effects on those targets. There's also the notion of autonomic tone, which is basically the volume or the degree of stimulation being supplied by any target, whether you're talking about the sympathetic or parasympathetic system. So usually both systems provide some degree of innervation or stimulation to their targets all the time. Okay? So there's some degree of sympathetic and parasympathetic tone all the time to all the organs. Okay? However, they don't equal in strength. The different tones are not equal. So under resting conditions, the parasympathetic tone to all targets of the body are much, much higher than the sympathetic tone. And it's under conditions where we activate some sort of stress response that changes, where the parasympathetic tone will decrease and the sympathetic tone will increase. So both are usually active all the time, but they are different in terms of how their particular tones and what dominates. So control of the efferent division of the autonomic nervous system, and that's really everything we've talked about. Control of the efferent commands going out to the viscera originate within the brain, and there are two parts of the brain that control autonomic function, and that's the hypothalamus and the brainstem. So within the hypothalamus, which is drawn here, we find that there are different areas of the hypothalamus that regulate different things. So heat control or temperature regulation is controlled by one particular part of the hypothalamus. Overall water balance, meaning the relative concentration of water to solutes throughout the body, is controlled by a particular part of the hypothalamus. And appetite and feeding, among other things. Other parameters of control are the responsibility of the brainstem, including things like urinary bladder control, respiratory control, cardiovascular control. Okay? So these parts of the brain are constantly receiving sensory information on all of these parameters. Okay? So through the autonomic afferent arm, all of these parts of the hypothalamus and the brainstem are receiving information on visceral function and then deciding whether or not an efferent command needs to be issued. Okay? So they are monitoring sensory input, and they are controlling efferent or stimulatory output to all the target organs. So this is something I composed in order to help you study the sympathetic and parasympathetic nervous system, and it just highlights some key things about each system. Things like the location of the preganglionic cell bodies, of both the sympathetic and parasympathetic nervous system. So where are the cell bodies of the preganglionic cells? Where are the cell bodies of the postganglionic cells? The relative length of the preganglionic and postganglionic fibers. Okay? The nature of the first synapse between the pre and post neurons, pre and post ganglionic neurons. Okay? In terms of what neurotransmitter is being released, what receptor that neurotransmitter is binding to. Also, the nature of the second synapse between the postganglionic neuron and the target cell, neurotransmitter and the particular receptor that it binds to. And then finally, in terms of, we are talking about the nervous system, but each system actually has an endocrine arm. And we talked about in the sympathetic system, that endocrine arm is through stimulation of the adrenal medulla and its release of epinephrine that binds to predominantly beta receptors. But what we didn't mention, and we're not going to talk about until later when we talk about digestion, is that the parasympathetic nervous system also controls the release of hormones. In particular, the hormones that control GI function and digestive function. And we'll talk about many of those when we get to that system. Okay, so use this slide and this table in order to help you study some of the key details. And generally speaking, all of the details that I'm listing here on this slide, I do expect you to know. Okay.